Well, good afternoon, friends and colleagues. I hope that you're having a great conference in Fortaleza. I was hoping to be there with you, but it was impossible for me due to some last minute circumstances to travel this time. So I have agreed, I offered, and the conference organizers agreed to keep me in the program through this recorded presentation, and hopefully I will be able to join you for questions on a video call after the playing of this talk. So technology has allowed us to not spare you from this modeling talk. I am aware that this will be the first talk of the day after the conference party, so hopefully I managed to still make it a little bit entertaining, although I was hoping for some more interactivity if I was physically there, but let's see what we can do today. So I have titled the presentation Mathematical Models of Anaerobic Processes, Tools for Technology Advancement and Scientific Discovery. What I had planned as agenda for today is to do a bit of an introduction. I always like to introduce modeling talks with some basic fundamentals in case there are people in the audience which are not familiar with models. And then I will start looking at the reasons why we do anaerobic bioprocess models, and then sort of show you examples of models that were developed for control purposes, decision and operation, and the models that were some models that were developed also to learn the process science. I will do this through examples of the work we have done through my PhD students over the years with collaborators. I thought that could be given a good sample of these specific cases for modeling, and hopefully you guys get excited and interested about models. So allow me to start with some introduction of modeling bioprocesses. So I always start when I talk about models with some reminders, so people have that always in mind when we are looking at what we're going to see later, right? And we understand the reasons why the models will look in that way. So the first thing we need to remember when we talk about models is that the objectives of that particular model that we are building are going to be the ones defining the type of model that we will have, as well as its level of complexity. What do I mean by the type of model is whether the model is going to be mechanistic, which means it's trying to represent natural laws or phenomena that we know and understand through the scientific knowledge that we have. And that typically happens by representing these physical, chemical, biological laws that we use to describe the system, things that are actually occurring mechanisms. The other type of models is the empirical models that we all know. The empirical models are just data-driven, in which we just use correlations of data, and lately, of course, with the advancements in AI and the computing power, we can do things like machine learning with neural networks, etc. So those models are basically descriptions of the system based on previous data, lots of previous data, and then they learn relationship between the data, but they don't describe mechanisms. A point about complexity is that the level of complexity that we will see in the models is always going to give us a trade-off between having a more accurate model and having a more difficult model to calibrate and simulate. So that's also something I would like to keep in mind throughout the talk when we look at the models we're going to be showing. And then I always try to remind this as well in all our minds, that the models are actually representations of the real system. So they are not actually the real system. So we cannot start thinking on the model as the actual reality. So that implies that all models are wrong. This is a famous quote. All models are wrong, but some models are useful. And also, do not fall in love with your model. That's another important thing. And if the model does not fit the reality, it is not normally the reality who's at fault. So that's also something we, as modelers, sometimes we need to be reminded of. And notice that I am not going to talk about AI in this presentation. So I'm going to focus all the presentation on mechanistic models, which is the main area of research in which I have worked for 20 plus years. I'm aware there is a lot of interest in AI. We can discuss a bit about this if you like during the questions, but mainly this talk is going to be on mechanistic models. So how do we do biokinetic mechanistic models in bioprocesses? Many of you are familiar with that. We typically use a balance. We decide which components, concentrations of soluble, solid components we are interested in, and we make a balance for each one of the components, in which we have the change of concentration over time, the influent concentration, the effluent concentration, and then we have some reactions which are occurring. Could be many reactions occurring at the same time. We put them in a matrix sometimes of a stoichiometry, which contains in columns the stoichiometries of what's reacting into what, minus for reactants, plus for products. And then we have, for each one of those, we have a rate, which we can put in a vector, and then eventually by multiplying the two, we end up with the net generation term of a particular component across all the processes in which it is involved. This is the typical way where ASM models and ADM models are built. This is typical from biokinetic models. How do we model the rates in the systems, in these models? Well, we typically use Monod or Herbert-Peart relationships, which are basically saturation, semi-empirical approaches for delimiting substrate into the rate, the specific uptake rate. We can have things like maintenance involved in there, and then we have yields. And when we have multiple substrate limitations, we typically multiply by several saturation terms. So we always have the contribution of the three limiting substrates. For example, this is for aerobic if it's also limited by oxygen or nitrogen. And then these models normally include some way of dealing with the inhibitions. If we have reversible inhibitions, typically we add a factor, which is going to go between 1 and 0. It's going to decrease our rate proportionally to the inhibitor. And then if the inhibition is irreversible, normally we deal with that as an increased maintenance or an increased death rate of the cell by adding a bigger value to that. Also, we can look at how do we represent the microorganisms in the model. If we look at the microbial population, sometimes we can have assume that all the biomass is the same and all biomass have the same composition. So we call all the biomass, we call it X, and we give it a composition, and this is my biomass. We could just do a model like this. Sometimes it's not, or a pure culture model will not require more complexity. Another alternative is to go on a more segregated approach. This is what we have in the ASM and in the ADM models, in which we have different microbial groups. So we assign differences into them. We might keep still the same microbial composition for them, but we differentiate between aerobes, nitrifiers, denitrifiers. In the case of AD, we can differentiate between acidogens, acetogens, and methanogens in different groups. So we have this level of segregation. Some models as well, they look into internal structures. So you can imagine a model, for example, with storage. If you have a model in which you are assuming storage components, so the biomass composition changes, so we could actually do a balance on the storage components. And then, of course, you can add a much more complicated model in which you differentiate between the biomasses and between the internal structures. Okay, so now, if we look at the anaerobic digestion system, the anaerobic digestion process from the perspective of a modeler, we can look at it with different lenses. An engineer can look at it as a process which is consisting on a slow step, followed by an intermediate fast step of fermentation, and then by another slow step of methanogenesis. So that could be like this three dynamics system, which has some inhibitions in the middle. So that could be one way of looking at the system, trying to understand and trying to control that behavior and maintain it stable. If we look into more details, we can have a bit of an understanding of the chemistry, the physicochemistry involved, the gas-liquid transfer, pH, alkalinity. That's a perspective. And then we can look at it from the microbial point of view. Many of you are microbiologists. So we'll look at it and in terms of what organisms are there, I mean, what kind of structures do they form, what's the community structure, the assembly, and of course, if we have granules, they are also very interesting ecosystems with gradients of concentration and competition for space and so on. So with that in mind, the question is what do we build anaerobic digestion models for? So I think we build models for two main reasons, and two purposes which are actually complementary. The first reason why these models were built was to make process decisions, which means we want to use the models to control the process, to design the process optimally, to optimize the operation at all levels, and then deal with instabilities, create all kind of tools to be able to optimize my plant, right? So that's a tool for the engineers, let's say. But the second purpose as well, which is complementary to that one, is that we can build models to learn the science of the process, to understand the mechanisms, constraints, and functional principles. So we can also try to describe these models at the level, let's say, in a lab, understanding what is the science behind the process. So those models will convert our data into understanding through the models. And then with those models, we can do actions in a process and in a real plant. So if we look at anaerobic digestion models, once we have these ideas, introductory ideas about bioprocess modeling and biokinetic models, what models do we have in anaerobic digestion? Well, in anaerobic digestion history of modeling, we have basically a world before ADM1 and a world after ADM1. So if we look at the world before ADM1, we have the first pioneering work, I think, on modeling anaerobic digestion was the work from Perry McCarthy. Perry McCarthy was just looking at the process, and by seeing what the process was doing, having probably very little knowledge at the time on the microbial composition, not even of the existence of archaea as such in those years. And he was able already to put together a very close to reality model at the time. So his work was truly pioneering. And then we have developments adding more complexity and understanding some more phenomena throughout the years, including solids degradation and including ammonia inhibition. So there was a few models popping up every few years in the world. So those guys are actually the pioneers upon which we build the subsequent models. So we can quote, you can use the quote from Sir Isaac Newton when he sent it to Robert Hooke, saying that if we have seen farther, is by standing on the shoulders of giants. I think Newton meant it with irony because he was trying to insult Hooke, but in this case we don't mean it with irony. We mean it really, we are standing on the shoulders of these modeling pioneers. And then ADM1 arrived. ADM1 was the result of the task group of anaerobic digestive modeling formed under the IWA auspice. And the report, the technical report, was led by Damien Batstone in 2022. It was published, and we know this model became the standard and the reference to consolidate a common language, a common biochemical framework, and allowed for a lot of benchmarking, and basically was extensively used and implemented in many platforms. The model is actually a very large model which contains 24 components, so it provides an actual dynamic simulation of 24 concentrations of soluble components, biomasses, and gas components. And it does it accounting for 19 reactions and conversion processes. All these steps in anaerobic digestion that we know about, plus the gas liquid transfers, etc., are somehow described there, which leads to a very large number of parameters. One of the strong things of the model is also that it has a very detailed ionic speciation and pH, so you can actually simulate pH, simulate all the alkalinity very accurately. That's one of the strong points of ADM1. So this model was a breakthrough, and it meant a before and an after for the modeling of anaerobic digestion processes, as we all know. The model itself has also limitations which are associated to the nature of the process and the complexity of the process. So the first one is it actually comes in with a fixed specific number of pathways which are actually rigid. So the structure of the model is what it is not allowing for new pathways in principle unless an extension is built. So this is missing alternative metabolism or co-cultures that could be present in an anaerobic digestion process. So this model will not be able to describe it. It also doesn't describe thermodynamics very well, which is relevant in anaerobic systems when they are running close to equilibrium. We'll talk about it a bit later with some of our modeling work. And therefore, it doesn't allow also for variable stoichiometry. We know the fermentations don't produce the same products all the time. If we stop methanogenesis in this model, we're going to have a constant stoichiometric coefficient of the acidogenic products. So you will always produce the same amounts of products no matter what. And we know that that is not realistic. We will see some work about that later. Also, as well, the description of disintegration and hydrolysis is not that detailed in the original version of the ADM1, which is a part that has been much improved later with the extensions. So the ADM1 created a breakthrough in anaerobic modeling, and then to address these limitations for the different cases and to expand its applicability, a lot of work was done on extensions and integrations of the ADM1. These contributions are on all kinds of areas of the model addressing these limitations. There are extensions adding biochemical pathways and adding thermodynamics. There are extensions improving the physicochemical inhibition processes, the precipitation processes that are not there. There are works extending the reactor implementation of the model to reactors with different biomass retention. Biofilms have been done. CFD has been done on ADM1 as well, and also its integration with other parts of the plant. And another area also has been done is application to non-methanogenic systems. So how can the model we use for fermentative non-methanogenic systems? So we have quite a few words on that area as well. So this has been actually busy after ADM1. The modeling community has been busy. There's a lot of literature, a lot of good work, and a lot of extensions that are available, so people can actually adapt the ADM1 to their specific process. If you have a process with sulfate, if you have a process with a particular type of solids, you can probably find a way around some extension for your application. So that brings me to the first case I wanted to mention. This is actually an extension that we built with the work with Wasim Ahmed, who was my PhD student here in Khalifa University before the merge. He was actually on the Masdar Institute with me. And what we tried to do in this work was to extend the ADM1 to incorporate sulfate reduction, which is a process which is not included in the original model. So to include sulfate reduction, normally we need to just add the components for sulfate reduction, the sulfate and the sulfide, and then those reactions will mean that the sulfate will be reduced by taking electrons from some electron donor. But we know that there are many BFAs available in the mix of an anaerobic digester, and we also have hydrogen. All of them are possible electron donors. So the question comes here with the trade-off between complexity and simplicity of the model. If we are building a model considering the 4 electron donors, we have to add 4 processes to the system with the biomasses, and then we have to calibrate the parameters for all of them and the inhibitions, etc. So that adds quite a lot of complexity. So what we wanted to do with this work is to assess what's the trade-off between the error we have in predicting the experimental data and the complexity of the model. And we tried the combinations of the one single electron donor, the case of hydrogen. Here on the left, we see the error that we have seen for each one of the models. The one on the right is a huge error if we only use hydrogen. For sulfate reduction, the model doesn't really work very well. But if we use acetate only, the results are already in the range of acceptability. The interesting jump, we see it by including acetate and hydrogen together. By introducing acetate and hydrogen together here, we have already almost the same error than if we keep adding propionate and butyrate with all this extra complexity. So the extra complexity doesn't pay back here. So the conclusion is that I think we can do a pretty good job just adding an extension of sulfate reduction with acetate and hydrogen. That was the conclusion of this work. Now, another work that we did, this was previous work I did in the University of South Wales with the student that I was co-advising, Bharat Penumatsa. We wanted to see if we could extend ADM1 for a non-methanogenic case. A non-methanogenic case meaning specifically for biohydrogen. It was of interest for biohydrogen production. And the idea was that they had experimental data in which they had the information about what products were being produced, what were the yields, fraction of products of fermentation, based on different conditions. And we saw that the main variable perhaps defining the most variability of these yields was the concentration of undissociated acids. So what we did is we created this graph on the left in which the stoichiometric coefficients of the acidification are changing with the concentration of undissociated acids. Therefore, we can run in real time. As the ADM1 is running, we always have the undissociated acid concentration. We calculate it and we use the corresponding stoichiometric coefficients for the fermentation. And that improved very significantly the predictions of the intermediates in the case, for example, of an overload. If we have an overload of the system loses its methanogenic activity for a while, and then we have a pure fermentation. So that actually has relevance not only for prediction of products, but also for the accurate prediction of an instability during anaerobic digestion. So we are very happy with this work. So now, these were two examples of model extensions, and now I wanted to enter already in the first main reason why we built models, which is building models for control. And I wanted to talk about a couple of examples from our work, again through my PhD students. So this is the work from by Santiago García Gen. Santiago was in Santiago de Compostela with us, and he was co-advised by myself and Juan Lema. And he did a great job actually in two aspects of this. So he did a first part of the work was to adapt the ADN1 model for co-digestion. So his thesis was on co-digestion, the application of modeling to co-digestion. And then when we have co-digestion, it means we're going to have very different substrates. And if we have only one substrate in ADN1 as soluble sugars, we are a little bit limited. So what we wanted is to incorporate how we handle when we have additional substrates that are soluble. So we did this modification in which we handle those as sugar equivalents, and we are sure that the electron balances are correct, so we don't have a deviation of the methane production when the substrates are more reduced or more oxidized. So that was his first piece of the work. So we have a model that can take different substrates which are soluble. And then the second part of the work is he did this work in collaboration with INRA in Jean-Philippe Steget's group in France for a few months, and he was modeling experimental data they had on the degradation of different agricultural solid substrates. Here we modify the model by adding a fast and slow carbohydrate biodegradable fraction. There was a significant difference between the two, so that added a lot of value to the model by being able to handle this hydrolysis in a more detailed way. Okay, and then what he did here is, once we have a model which is able to handle co-digestion, then we can use the model for control design and purposes. And then what he did is two things. The first one was to design an optimizer of the blending of the co-substrates that we are going to be using. So that was done by using the information on the biomethane potential test that we can do from each one of the substrates based on how much methane is obtained depending on the time, the residence time. We can add that into a linear programming optimization problem to maximize the methane production. So this is going to suggest a blending of co-substrates for maximum methane production. And then because we have a great model, the anaerobic digester here is our great model already expanded and adapted to capture these complexities of the substrates, we can simulate what's going to happen, and then based on the flow of methane that the process is producing, and ADN1 also predicts very well, as I said, pH and alkalinity, so we also get the alkalinity ratio and methane flow rate, which are two indicators in anaerobic digesters good performance. So those two are actually used to diagnose the stability of the process for the hydrogen and the remaining capacity that we have by looking at the methane production. So that allows us to establish a control strategy which aims at pushing the reactor farther if we still have methanogenic capacity and we are in a stable condition, so no hydrogen spike. We can push the reactor farther. How can we push it farther? We relax some of the constraints in the optimization of the blender. For example, we allowed for a substrate with more capacity for methane as long as the reactor is not being upset. So we'll have another loop of a new blending of substrates to keep pushing the reactor. So this is a direct application of a model for control, and I think the work of Santi is clear representation of this. I think one of the main outputs was also that apart from the publications, this has been patented in Spain, and I think the patent has also been licensed to a company. The second application of models for control for control design that I wanted to highlight is the work also by Wasim. It's the second part of his thesis that he worked on with me in Khalifa University. to use model predictive control. So model predictive control, for those not familiar, I just introduce it briefly here. Model predictive control, what it does is that it measures the current situation of the process, and then it simulates in the future alternatives, the path that we can follow. For example, in this case, we're going to start up an anaerobic reactor, so we could just try different flows, increases of the substrate, maybe dilution, maybe addition of alkalinity. We can combine those flows over time and project with the model into the future what's going to be my development of the methanogenic capacity in the reactor. So in that way, what we can do is to try to follow that projected optimum for a while, and then we take another measurement, and then we start again, and we look at, okay, we were supposed to be here, this is where we are, now we re-optimize again, and we continue to try to follow that optimum path. So we apply this work for the automatic startup of an anaerobic digester. As you know, anaerobic digesters are started in a very manual way or empirical, so people apply a percentage of increase on the organic loading rate every few days, and this is done, in our opinion, that was suboptimal. It could be done better. So what we wanted to build is we wanted to build a very simple model that contains only variables, that requires only inputs that people are actually measuring in real plants. Because if we are trying to simulate with a very complex model, the MPC projection, we need to feed it a lot of information, and in most plants, when people are starting up the process, they are not measuring 20 different variables, so those was not feasible. So we wanted to build a simple model. We built a model in two-stage acetogenesis, which required only to tell the model, okay, what's going to be the flows we are going to apply, and what's going to be the concentration of acetate in the system, the methanogenic, acetoclassic methanogenic activity, and the methane flow rate. With those measurements, we can tell the model how we are, and we can project a simulation into the future following a path of flow rates increase, and see how that methanogenic capacity is going to increase. We are measuring the startup of the reactor as an increase in the methanogenic capacity. The model was contained in the equations that you see there, which I'm not going to detail. And this is the architecture of the system. So we have the model predictive controller is actually supplying some programming of flows for start up the system, and it's checking what's the projected future of this, and it keeps optimizing until it finds the optimum way according to this predictor model, and then it applies it to the real plant. Of course, we didn't have a real plant to play with this, and here it is where it comes very handy to have an ADM1 based virtual plant that we can use to test our controller strategy that would be in reality applied in a real plant in the future. So that's what we did, and we have different startup strategies showing here, manual increases of different percentages, but the solid black line is our best MPC, and you can see that is the fastest one in achieving methanogenic capacity, and is the fastest one in achieving methane production rate. This is the acetoclastic methanogens, and this is the methane production rate. So having a model predictive control provided us a strategy for optimization of the startup automatically of an anaerobic digestion. So this is a second example of the application of anaerobic digestion models for control. All right, and then hopefully we have an idea, or at least I'm convincing you that these models are very useful, because we can design controllers using them to test those control strategies. However, the second application, and this is one that I am particularly interested on, is to make models to learn the science. And I want to talk a bit about this now. So if we think about the biokinetic models, the models we have been discussing now, like ADM1 model, that model has itself, those kind of models have some limitations. The limitations are coming mainly from the idea that they're not really very suitable to include variable metabolisms or changing pathways, because we have a constant stoichiometry that we multiply by a rate, and this is normally the way these models are built. There are ways around it, but they are fundamentally not, they are kind of patches, like the one I showed you before with the variable stoichiometric yields there. There's also limited description of ecological interactions by these models beyond substrate competition. So if there are other interactions in between organisms, this is difficult to account for it. One of the main drawbacks of biokinetic models is the great difficulty to estimate those parameters, the Ks for the monods, the maximum specific uptake rates and the biomass yields separately is a very hard one to identify, because we don't normally measure biomass groups separately. We measure total biomass, and then we observe mu max, not Q max and the yield separately. So that's a difficulty. And in the case of KS values, it's really difficult, because the only real way of identifying properly a KS value is in steady state chemostats. And what we see in reality is that people are running plants, and then we have the literature plagued with reported KS values and combinations of these parameters, which are sometimes orders of magnitude different from each other for the same process, which tells us that these parameters are not really identified, because we end up having infinite combinations of parameters which are given the same goodness of fit. So what one goes up, the other one goes down, and then we have everything in between of the values between them too. So this is a big drawback of biokinetic models. The main other drawback is that these models are not very powerful to learn much about microbial ecology. We cannot integrate there also things like molecular data that we have now. So what alternatives do we have? And this is where a big area of my research since my PhD time comes into play. And then I always like to describe this with an example which I'm using is related to Formula One driving. So I like to ask whether we could describe a microbial process function without actually knowing or understanding completely the microorganisms that are there and their genetics and their metabolics and everything like this. And I use the analogy of the Formula One drivers. So if we think about the Formula One driver, we can model the guy, model his brain, what's he or she going to do driving the car as fast as possible. But we can also solve very easily in engineering. We can calculate with mechanical engineers and on some dynamic programming problem. We could solve what would be the optimal movements, acceleration, braking, turning, and so on, in a particular circuit. That can be calculated mathematically through models. So if we calculate that with those mechanical models, which are not that complicated, not as complicated as a person, we can actually get a prediction of what the drivers could be doing. But if we actually overlap that with what the drivers are actually doing, the predictions are very close. So these guys are actually driving very close to the optimal. Why are they driving very close to the optimal? And why can we predict that from them? Because if they don't do that, they would not be Formula One drivers. They would not be playing. And that concept can be applied as well to microbial ecosystems. If we can define and calculate what are the optimal performance, or what's the best activity or metabolic products or function that a microorganism is going to have, we could say that most likely this is the one that is going to have, otherwise the organism will be suboptimal and not present in the system anymore. So the idea is that whether we can predict and direct the performance of a microbial community with little understanding of its structure. And that's a principle we try to follow to predict the products in anaerobic fermentations without going into the microbial composition. This is the work that started during my PhD, and then Rebecca González Caballero, which was our student in Santiago, together with Juan Lema, he continued working on this and developed the work further. She's now assistant professor in TU Delft in Netherlands. So she worked on predicting the product formation in anaerobic fermentations, but these are non-methanogenic anaerobic fermentations. So the question for us was here, can we direct the production of these fermentations towards the products that we are interested in by changing things like pH, substrate concentrations, hydrogen pressure, things that we could manipulate. For that, we need to first understand and model what these organisms will be doing depending on the conditions. So we apply this approach to model the systems in a very different way than the biokinetic models. We did it through bioenergetics. And now, before that, I want to do an intermezzo to introduce some concepts about bioenergetics, because I'm going to be using them later. So to keep you on board and avoid losing many of you in the process. So the first thing I want to remind, most of you will know this, biologists, maybe some engineers need a reminder, is the way that cells microbial cells will harvest energy. So there is chemical energy in whatever catabolism they are doing. The cells can only harvest energy if they make ATP. And they can make ATP in two possible ways. One of them is by coupling an ATP production to a specific reaction, substrate level phosphorylation. And the other one, as we know, is through the chemiosmotic proton translocations. So some reactions, if they're happening in the membrane and they have a surplus of energy, they can be coupled to a proton translocation through the membrane, and those protons are coming back through the ATP synthase. Every 3 protons, more or less, makes an ATP. So that allows for life to really run in very low energies metabolisms, because the quantum of energy, minimum quantum of energy that the cell can recover is actually a proton translocation, which is about 1/3 of an ATP. Great, so that's how cells harvest energy, but the energy needs to be there in the first place. So you have chemical energy, you have a reaction like this one I'm showing here. How do we calculate how much energy is there? We all know that from thermodynamics in undergrad. So let's say we have a combustion, oxidation, if you like, of glucose. So we can calculate the delta Gibbs of the reaction by putting the values of the formation and calculating the difference, right? Products minus substrates. And if we do that calculation, we know that glucose is close to 3,000 kilojoules per mole of glucose oxidized. That's the combustion of glucose gives energy. Instead, if we do anaerobic methanization of glucose all the way through all the steps from glucose to methane and CO2, all the energy that is available there is only 428 kilojoules per mole. And this is in standard conditions. So we can calculate things like this for any reaction that we're interested. But these values that we get are actually in standard conditions. The problem is that I don't know about you, but I have not seen many reactors in anaerobic systems which are working at 1 molar concentration of all products and substrates and 1 bar of gases. So the reality is that we don't work close to standard conditions, we work far from standard conditions. That means the effect of concentrations of products and substrates modifies the Gibbs energy. How significant is that? If we have 3,000 kilojoules, it's not very significant. But if you have 70 kilojoules, for example, for propionate oxidation, which in standard conditions is actually not running, if you have propionate oxidation to acetate and hydrogen, is a plus 76 kilojoules per mole. That reaction doesn't run. But of course, as we know, in methanogenic environments, the hydrogen pressure is very low because we have methanogens harvesting the hydrogen. So the pressure of hydrogen can be very low. If we put the pressure of 5 Pascal and everything else in realistic millimolar concentrations, the reaction actually can have, in this particular case, negative 18 kilojoules. So the reaction can run now. So that's how we calculate these stories. So what we did for the fermentation pathways is we took the entire acidogenic pathways that are known. I mean, not the entire of them, the most important ones leading to the main products, acetate, propionate, butyrate, and we describe them step by step. And we identify in those reactions in which places energy is harvested by the cell, by SLP, and also in which places energy is harvested by the cell by proton translocations, potentially. Some of those proton translocations are linked to transportation of substrates outside and inside the cell, and we calculate this, and that is how the effect of pH comes into place. If you have an acid and you want to transport an acid into an acid environment, it's going to be less energetically interesting, and if it's an alkaline environment, it's going to be more energetically interesting. We analyze those thermodynamic pathways, calculating how much energy is in each one of the steps, and at the end of the day, what we did is run an optimization problem of the fluxes in these pathways, assuming that the cell will do whatever is the optimal for the maximum energy. And that will lead to a set of product formation. And this is how we predict the yields that the metabolic network of reactions and organisms would be doing for a particular condition. For example, in this case, I'm showing data results for pH. And up there in the slide is the experimental results from Temudo, and these are the results from our predictions, which obviously is not a direct fit, but is already showing the main trends. And the interesting thing is that this prediction is coming from first principles, the first principles of just applying balances, thermodynamics, and optimality. Okay, then the last part I wanted to discuss is a bit the work we did on the pathway analysis, a more detailed pathway analysis in microbial systems, which is the work that we did through two of my PhD students, Mauricio and Ahmed. Ahmed's now with us as a postdoc. Mauricio is working in another department here after he moved into epidemiology, after the work we did for COVID. So both of them work on this area. First was Mauricio, and then Ahmed followed up on it. And the reason why we did this work is to try to connect the top-down approach that we have in bioprocess, the development of bioprocess, in which people start with mixed inoculum, and then you apply conditions in a reactor, and you end up with a community that you enriched for a particular function. And there is this other approach, which is the bottom-up approach, in which you can actually select specific strains and then, optimize them for a particular product, and then just design your culture for a particular high-value product. So those two approaches somehow are meeting in the middle, and we want to help bridge that gap with this work. In this work, we try to analyze the pathways in detail. So we pick a pathway of interest, and we try to understand where are the bottlenecks of the pathway, and how are the trade-offs between rate and energy in the pathway. So what we did is we actually picked a pretty difficult one, the propionate oxidation to acetate and hydrogen, which is an extremely low energy pathway. So it's a very tight pathway in terms of energy restrictions. And we coupled that to methanogenesis, which I will show the results in a bit. So what we did is we look at the pathway. What is a pathway? A series of reactions, as we know, steps, and we did a catalog of all the steps that are involved in all pathways that we could find in the literature for propionate oxidation to acetate. So propionate can move to acetate and hydrogen through different alternatives here. So we did an inventory of those, and then we also took into account the role of electron carriers. We know that some steps in the pathways are oxidations and reductions. So those oxidations and reductions are conducted through an electron carrier. The electron carriers, what's the importance in having different electron carriers is that they hand in the electrons at different energy levels. It's like the electron is transferred into the substrate in a reduction, but the energy level of that electron is different if it comes from an NADH or if it comes from a ferredoxine. So that has an important role in the energetics when we calculate them. So what we did here is we did an inventory of these main 6 pathways, and then we looked at the variants or different configurations of the pathways in terms of what carriers could be used in each one of the steps. When we have an electron carrier here, that means it's an oxidation or reduction. So what choice of carriers we are allowed based on the literature, biochemically. So that creates a lot of combinations. If you can imagine now, we have a pathway, but if you start doing all possible combinations of carriers in all the pathways, you create a big number of combinations. And then we also have choice in terms of how many protons the cell is investing or translocating through the membrane in some particular steps in which energy is needed or energy is available. So if we allowed for all that combinations, this led to us to have about 500,000 combinations for a particular condition of pH, temperature, and concentration. So that looks like a big number, but it's computationally very doable to evaluate. And then what we do is for those combinations, we evaluate the feasibility of these combinations, the different variants, and we calculate how much energy they make for the cell in terms of ATP. Why do we do that? Because we want to find the optimal one. Which one is the best pathway configuration that is going to lead to the most energy yield to the cell? And that hopefully will help somebody developing a pathway from scratch to actually follow that optimal pathway. If you want to make a synthetic organism that is going to survive in an open ecosystem later, ideally we would like to develop an organism that is as optimal as possible, because you can modify the pathway for your high value product, but the moment you put it in the wild, the organism is not going to last very long. So we hope that this kind of work will inform the bottom-up approach by designing the pathways following the optimality. So a pathway to be feasible needs to have 2 things. It needs to have a Gibbs energy that is not positive, and it needs to have concentration of the intermediate products that they have to be within reasonable range. If I have to lower my product concentration to 10 to the minus 12 to be able to run that step in the pathway, obviously 10 to the minus 12 is not a feasible concentration inside the cell, because we will have not a single molecule inside the cell. And then if in a pathway I can find a way in which all the Gibbs energies are negative and all the concentrations remain within the range, then that pathway is feasible, and then I can just count how much ATP it makes for me. And we try that for different environmental conditions, and we can just look at the solution. For example, for this condition, we see in this particular condition the only pathways that made any ATP for the cell were the ones shown here, and how much ATP it was actually made for the cell. The other pathways were actually making no ATP or were basically not feasible in this particular condition. And we can try different conditions. For example, here this shows results for different hydrogen pressures. I think is the most interesting. You can also look at the others, but the hydrogen pressure is the most interesting. You see how as the hydrogen pressure increases, we have less and less energy available and less pathways feasible. Our model also shows us what happens inside the pathway. So if you want to look into a pathway, it tells you what are the concentrations of the intermediates. And as long as they become within this range of -2 and 10 to the -6, the pathway is feasible. And you can see when they are getting close to the limit, it means that we have kind of a bottleneck there. So things are getting harder. So if we push it, if we increase the hydrogen a bit more, probably this guy is not going to run anymore, this pathway. And then here, for example, we have an accounting of how much energy in each step is being invested or harvested by the cell. Here's an ATP. Here is investment of protons and release of protons, so we can account for how much energy the cell is recovering. And the yellow thing at the bottom tells me the loss of energy dissipation in some of those steps. Some of those steps will lose energy because they have too much, and that energy that is not harvested will be lost. That will be actually the driving force. So we did the same thing for methanogenesis, and we put them together. And then in this graph, which is kind of a scary graph, what we're plotting is the hydrogen pressure increasing towards the right, or any other equivalent to that. So dissolved concentration or partial pressure, or they're all the same thing equivalent. Even formate, if you think hydrogen is not the syntrophic electron carrier, you can use formate, or just volts if you think the electrons are just transferred directly to each other. So as we increase the hydrogen pressure or the voltage, what we see is the propionate oxidizers, they have less and less energy available, obviously, because the hydrogen is higher. And then some pathways are still making some energy for the cell. And eventually, at 10 to the -8, the next one is already having no pathway that produces any energy for a propionate oxidizer in terms of ATP. If we look at the methanogen in the middle, it's the opposite thing. The moment the hydrogen increases, the methanogen will be able to make more energy. And then the interesting thing here is that they only overlap, meaning both of them can be having a leaving in terms of some ATP for everybody in a very narrow range around 10 to the -8 concentration of hydrogen or 1 Pascal. That is a value which is, in our opinion, very small, because 10 to the -8 is actually without this range of feasibility that we said. So obviously, as syntrophism science is saying that perhaps there are other mechanisms more complicated than dissolved hydrogen to transfer electrons between syntrophic populations. Probably this result is also indicating the same. Perhaps it's formate, which could be in the range of 10 to the -6, or perhaps the electrons travel in a different way. So with this analysis, we were able to explain a bit this phenomena in a more detailed way inside the pathway. And then, just to continue this work, I will not go into the details, but we are now introducing the importance of the driving force, because one thing is to be very efficient for a pathway, so you harvest all the energy. But if you don't dissipate or lose any energy, you don't have a driving force to run the pathway. And we know that this flux force efficacy means that if you have an enzyme that is reversible, and the driving force is very little, the backwards reaction is going to run almost as much as the forward reaction. So you are wasting half your enzyme doing the work in the opposite direction. So you need more enzyme in the cell to push this. So this is actually an inefficiency. It's a resource allocation problem. We want to work towards describing models to help us introduce that. For that, what we did is an analysis of these driving forces. With Ahmed's work, he develop a more advanced optimization system in which we have this trade-off between the driving force. So we are forcing to have a dissipation to the reactions, and the reactions will run at the smallest driving force. So here in the left, you see that the yellows are not 0 anymore, so they are all the same, and some of them are bigger. So reactions will run at the rate which is going to be depending on this driving force. So if you have a very small driving force, that step will be very slow. So we try to optimize this, and we see clearly a Pareto front in the different pathways of propionate oxidation. So if you want more yield of ATP, you're going to have less minimum driving force, meaning a slower reaction. So we have this Pareto front, and we can actually describe this Pareto front between the rate and the yield with this modeling. All right, so this is pretty much what I was hoping to show you today. And if I have to say some take-home messages here, I would like to just pick those 2 ideas that I wrote there. Biokinetic models, as you have seen, they are very powerful to control and to operate anaerobic bioprocesses. ADM1 is the proof of it. It has been used extensively, is implemented in commercial softwares, is used to control plants, and there's a lot of extensions available that people can plug in the model to their particular problems. So biokinetic models are useful for control. That's my first message. The other message is that also these models are not that powerful to learn the science. So they're not very helpful to teach us things about the process, new things about the process. And then perhaps we can use alternative mechanistic options, including metabolic models, bioenergetic models, or ecology models that are available there, and we showed some examples of some of those. So I like to send the final message that the mechanistic models can help us learn and create new hypotheses to advance the science. Why I'm saying that is because now, of course, we are in the years of the AI explosion, which means that AI will have all the answers for us. But I think if you just rely on black box models, which have learned from data, we don't have any certainty of what's going to come as an outcome is going to be actually the right one. And also we're not learning much, so I'm still, advocating for the use and not abandonment of mechanistic models. And I wanted to finalize just acknowledging the people I worked there on anaerobic process modeling over the years. There are my mentors and my collaborators. All of you, I want to thank them. And below there, these are my PhD students. So there's been a transition between my mentors and my PhD students with this work, which I'd like to thank them all for teaching me, and also for both of them for teaching me and for working together over these years. And I always like to end with this quote that some of you might recognize. We choose to do modeling of AD and other things, not because it's easy, but because it's hard. You might recognize that quote from President Kennedy for the moonshot. So with that, I leave you, and hopefully we can have some questions if I'm able to connect online. Thank you very much.